So now that we've looked at viruses and a little bit of other types of invaders that try to kill us, it's good to know that our bodies are capable of defending against them. So we need to look at some general vocabulary. First of all, a pathogen is any agent that can infect a host organism. We use agent here because it's pretty safe to say that viruses are non-living. So with pathogen, we mean any kind of parasite, living or non-living. Right? Anything that can still reproduce, though. And an antigen is any molecule or molecular shape that triggers an immune response. We'll see that quite a bit later. Quick overview of the immune system. This is the body system that recognizing antigens and then responds to them in such a way to clear it from the body. Innate immunity starts working immediately to protect the host. And so we're thinking this is like the most complex thing that you're going to get in plants or invertebrates. And then the adaptive immunity is slow but very targeted. So you will find these in vertebrates and higher mammals and so forth. And so we can look at it this way. The innate immunity, that's going to be working right off the bat. Now I'm going to tease out the barrier defenses kind of as their own thing and just look at these as innate, but you can, however you think of it is fine, right? They're really quick, but they're not very specific. The adaptive immunity is going to look at very defined chemical patterns that trigger a response. Okay, and in terms of the adaptive immune system, we can look at the humoral response and the cell-mediated response. So let's start by looking at barriers. All animals, all plants have barriers to try to protect against pathogens. In animals, we're thinking things like skin, mucous membranes, and bodily fluids. So our entire bodies are covered with skin, and anything that has to be open and can't have skin there because it needs to allow for the diffusion of materials, for example, your digestive system and your respiratory system and your excretory system and reproductive systems, those all need to have a way to get something from the inside out or vice versa. And so you can't just have skin covering the mouth, then you can't eat. That's really sucky. So your skin's super inhospitable. Dead cells, dry, low pH, it's the ideal covering. But in those places where you can't just have skin covering up, you're going to have a mucous membrane. And so mucus has all kinds of enzymes that break down microbes. It's also going to trap the microbes and allow you to kind of cough them up or wash them out somehow. And then our bodily fluids are, again, really inhospitable. For example, your tears and your saliva are going to contain enzymes that break down microbes so that anything that does get there is either washed away or killed. In plants, we can think about the fact that they have really tough cell walls, and in fact, infection is only going to ever occur in plants if there's some kind of mechanical damage that breaks away the cell wall. So for example, when you're harvesting certain crops, you can introduce mechanical damage, and that can allow pathogens in. There's a lot of different insects that will eat away at parts of the bark of different trees, and that mechanical breakdown of the cell wall can let bacteria or viruses in. Okay? All right, so now switching gears to the innate internal defenses. So once a potential pathogen enters, invertebrates can still do quite a few things to stop progression and to stop the establishment of an infection. They've got digestive systems still with lysozyme. Lysozyme is an enzyme that breaks down bacterial cell walls. They have cells that go around and just phagocytize anything that is foreign. So they ingest it, digest it, and pop it out the other side. And they also are secreting all kinds of antimicrobial peptides or little protein fragments that are going to interrupt the fungal or bacterial cell walls. So this is a very, very basic innate defense. If we take a look at vertebrates on the flip side, we're introducing some more highly evolved structures such as natural killer cells. These cells are going to be able to target infected cells or cancerous cells and cause their apoptosis to occur. There are interferons, which are just chemical signals, and that's kind of like a warning call to all the local cells that there might be viruses present. So go ahead and, get ready to attack. And then we have our inflammatory response, which despite the pain that it causes, does a number of things to help us out. So we're going to be thinking about releasing chemical signals. That's going to cause all the neighboring cells to increase blood flow to the area, letting those blood vessels leak a little bit and allowing white blood cells to get into the potentially infected tissues, and increasing either local temperature or systemic temperature. If it's just local, right, you stubbed your toe and you go to feel it and it's super hot, that's just a local temperature increase, or you can have systemic fever where your entire body gets hotter. Okay, so these are just invertebrates in higher animals that have a backbone. So the first three, those are in most animals. The later three are only in more advanced vertebrates. So we need to take a look at what the white blood cells do. I don't think I even knew most of this stuff until college. So essentially we've got this stem cell right here, this undifferentiated hematopoietic stem cell, and that's going to differentiate into every type of blood cell. It's going to make the platelets. Those are what help with clotting. It's going to make the red blood cells or erythrocytes. That's involved in oxygen movement. And then it's going to produce all these guys. So the basophils, neutrophils, eosinophils, monocytes, and natural killer cells. You do not need to memorize these. I'm typically just going to refer to these four guys right here as phagocytes. They're involved in ingesting stuff, right? So these are the phagocytes. The natural killer cells, it's neat to know about, but I doubt you'll see it on the AP exam, unfortunately. And then the lymphocytes we're going to talk about next, the T and B cells. So all of those are coming from one type of cell, and you can see that all of these guys, these are all what we're going to call the white blood cells. They actually are pretty darn plentiful. Okay, so how do phagocytes, basophils, neutrophils, eosinophils, monocytes, how do these guys right here that I'm now calling phagocytes, how do they recognize pathogens? Because that's going to be the first thing they do. Remember, we said that a phagocyte is going to come around, find a pathogen, ingest it, digest it, and spit it out the other side. Well, they do it with something called toll-like receptors, or TLRs. This is why it's important that you understand the differences between the different pathogens, because TLRs are each going to recognize a particular molecular pattern that is very specific to something that you would not find in the host. So it's specific to the pathogen. For example, single-stranded DNA or double-stranded RNA. We're just not going to have them in our bodies. We shouldn't. And if we do, it's probably a sign of something invading us. Peptidoglycan and a number of other chemicals, chitin and other types of sugar protein moieties are going to be really common in pathogens, but not common in us. So these receptors that are on the outside of the phagocyte are going to bind to whatever it is, and then as soon as that binds, it's going to cause the endocytosis of that material and then the breakdown of the material, right? So that's going to get rid of a lot of pathogens. But this is where vertebrates really shine. This is where mammals are amazing. We have an adaptive immunity. And what our adaptive immunity is it's like a system that really just targets exactly what you need to target, right? Let me rephrase. So this is going to rely on the innate defenses to recognize the pathogens, but now we're activating lymphocytes. Sorry, I'm moving around here. So lymphocytes are your B and T cells, and we've got millions of them. And each B and T cell has its own combination of molecules that can recognize a ton of different stuff. So we're going to call these guys right here in purple the antigen receptors. B cells have the ones that look like little Ys. T cells have ones that are just straight up and down. And this little notch is always going to recognize something different. So we have a whole arsenal of B cells, each with different antigen receptors. We have a whole arsenal of T cells, each with different antigen receptors. And they're each going to bind to a different, what we're going to call now as an epitope, or part of the antigen, right? So an antigen can be pretty big. These receptors are just recognizing little pieces of them. Now, it's important here that a B or a T cell that recognizes something that is normally present be killed or somehow removed from the population before your immune system really goes into play. So before birth, each one of these millions of B and millions of T cells are checked for self-reactivity. If they do cause a reaction in your body to something that is not foreign, then those are like silenced. Okay, so for the adaptive immunity, we're looking at two kinds. The humoral immunity, or the humoral response, involves B lymphocytes, and those are going to respond to antigens that are in the blood or other body fluids, right? Your lymph system, etc. So humoral is referring to the humors or the liquids of the body. And what's going to happen is the B cell is going to bind to the antigen, and that's going to cause this type of B cell with this type of very, very specific receptor that bonds to this particular epitope right here. Notice this whole piece is an antigen, but the part that it's binding to is the epitope. So now we're going to take this B cell, and because it matched, it's going to go through a really rapid cloning phase. And that's going to cause the development into two types of cells, either mature active B cells that are now no longer going to have the whole antigen receptor. They're just going to release part of it. That's what an antibody is. It's just part of that B cell receptor. So the mature B cells are either going to be responsible for like releasing the antibodies. Antibodies can then neutralize whatever the pathogen is, or they're going to make B cells that are part of your memory B cells, so that we'll talk about those in a second, actually. Next one. Okay. Our cell-mediated immunity is involved in killing our own cells that have been infected by a virus or a small bacteria that is an intracellular parasite. So in cell-mediated immunity, we're going to be using the T lymphocytes. What these guys are going to do is they're going to recognize antigens that are presented on other cells. So this idea of antigen presentation is really important. They're not directly binding to the antigen per se, but to something called an MHC complex. You don't need to know about this. Essentially, they're going to respond to other cells that are like, hey, look what I found. So it's either going to be a phagocyte, right? So it's digested something and recognizes, let's say, a virus. The phagocytic cell is going to ingest that viral particle and then take the various pieces of it and show it on the outside to say, hey, I found this guy, he's a bad guy, break him down, okay? Or infected cells can do the same thing, but instead of kind of giving a warning like, hey, keep an eye out for this guy, the infected cells are going to present and say, bad news, I'm infected. And that's going to cause the T cell to bind, release all kinds of chemicals that cause the degradation of the infected cell, and then like eat out of there so that it can find some more that are infected. That's stopping the viral life cycle in its tracks. It can't continue to divide in a dead cell. Okay. So just like when we saw the B cells, as soon as the T cell recognizes something with its receptor, it's going to go through really rapid division. So now let's say you have an infection of the rhinovirus, the cold. What's going to happen is the T cells that bind to that are going to rapidly multiply. And then anytime they come across a cell that is infected, it's going to kill it, which is delightful, right? Okay. So I kind of already talked about this. We're also going to be looking at memory cells with T cells as well as B. Now, an interesting kind of third category is what's called a T helper cell. It's a type of T cell. It's activated the same way via antigen presentation, but in this case, the T cell acts to work with both the B and the T cells. So what we really want to do with an infection is make sure to get the T cells activated. If we can activate the T helper cells, then we can get the humoral and cell-mediated responses going, because we're going to activate some B cells and make some antibodies. We're going to activate T cells, turn them into cytotoxic T cells that then kill infected cells, and we're in good shape. So one more video for you. The next one will be short.